Welcome to part 2 of Antimicrobial Drugs, General Considerations. Today, we're delving deep into the world of antimicrobial therapy, exploring the challenges and complexities faced by healthcare professionals when using these powerful medications to fight infections. Let's start by talking about toxicity. Antimicrobial drugs can sometimes cause irritation, either at the spot where they're applied or throughout the body. This can lead to pain, inflammation, or even abscesses. It's important to understand that this irritation can happen locally, right where the medication is used, or systemically, affecting the whole body. Systemic irritation can cause problems in various bodily systems like allergic reactions or toxic effects on organs. Next, let's discuss hypersensitivity reactions. Almost all antimicrobial drugs have the potential to trigger immune responses in unpredictable ways. These reactions can range from mild rashes to severe, life-threatening conditions like anaphylactic shock. Some of the main culprits for these reactions include penicillins, cephalosporins, sulfonamides, and fluoroquinolones. Moving on to microbial resistance, we encounter two main types: natural and acquired. Natural resistance refers to inherent traits in microbial populations that protect them from specific antimicrobial agents. This could be because they lack the target sites or metabolic pathways targeted by the drug. On the other hand, acquired resistance happens when microbes adapt over time to become resistant to drugs they were once susceptible to. This is often driven by the selective pressure exerted by the use of antimicrobials. Resistance can occur through mechanisms like mutations or the transfer of resistance genes between bacteria. Resistance can arise through mutation or gene transfer mechanisms. Mutation involves stable and heritable genetic changes that occur randomly among microorganisms. Over time, mutant cells emerge within a sensitive population, requiring higher concentrations of ALMA for inhibition. This gradual replacement of sensitive strains by resistant ones characterizes vertical transfer of resistance. Examples include single-step mutations conferring high resistance, such as enterococci to streptomycin or E. coli to rifampin, as well as multi-step mutations leading to gradual decreases in sensitivity, as seen with erythromycin or tetracycline resistance. Horizontal gene transfer, also known as infectious resistance, involves the transfer of resistance genes from one microorganism to another. This mechanism enables the rapid spread of resistance among microbial populations. Conjugation, transduction, and transformation are common mechanisms of horizontal gene transfer. For instance, conjugation facilitates sexual contact between bacteria, allowing for the transfer of resistance genes, such as penicillin resistance in Haemophilus or gonococci. Transduction involves the transfer of resistance genes via bacteriophages, with examples including certain strains of Staphylococcus aureus acquiring resistance through this mechanism. Transformation, although less clinically significant, also contributes to horizontal gene transfer by enabling resistant DNA to be taken up by sensitive organisms. Cross-resistance is another important aspect to consider. This occurs when resistance to one antimicrobial agent confers resistance to another. For example, bacteria that develop resistance to a certain type of penicillin may also become resistant to other penicillins, making treatment more challenging. Superinfections are another concern. Using antimicrobials for a long time or having a weakened immune system can disrupt the balance of microbes in our bodies. Conditions such as diabetes and disseminated lupus erythematosus can also predispose individuals to superinfections. In diabetes, high blood sugar levels can weaken the immune system, making it less effective in fighting off infections. Disseminated lupus erythematosus, an autoimmune disease, can also impair immune function, increasing the susceptibility to infections. Individuals with these conditions may be more prone to developing secondary infections, highlighting the importance of close monitoring and appropriate management to prevent complications. Antimicrobial therapy can also affect our nutrition by disrupting the balance of good bacteria in our gut, making it harder for us to absorb important nutrients like B-complex vitamins and vitamin K. In summary, antimicrobial therapy is a complex field with many nuances and challenges. By understanding these complexities, healthcare professionals can optimize patient outcomes and mitigate the risks of antimicrobial resistance. If you found this information helpful, don't forget to like, share, and subscribe for more insights into healthcare and medicine. Thanks for joining us on this scientific journey, and until next time, take care.